Hi, I'm Jennifer Katcher, and I'm a biology teacher at Pima Community College, and I'm here today to show you the steps of PCR, or polymerase chain reaction, that we're going to use as part of our DNA barcoding project. So, in a previous video, we extracted DNA from an insect, and you may have done that step, or you may be receiving DNA from an insect that has already been extracted, or you may be doing this project virtually. The first thing I want to show you about PCR are the components that are required to conduct the PCR reaction. So PCR is going to make lots and lots of copies of a specific section of DNA. So we have to have certain components in our PCR reaction that will allow that to happen. So I'm going to tell you about each component in the reaction and its role. So the first thing that we need, of course, is a DNA template. So that's the DNA that was extracted from the organism that you are trying to analyze. The second thing that is going to be present are primers. And for this sample that we're going to do, we're going to have forward and reverse primers. And what those are short segments of RNA that are going to have complementary base pairing to the DNA template. And it will tell the DNA polymerase where to begin copying the DNA. So we're not interested in copying the entire section of DNA. For our barcoding project, we're interested only in copying the region that is coding for the CO1 gene that is involved with cellular respiration. So the next component is one that I already mentioned. It's the DNA polymerase. And because our samples are going to be heated to relatively high temperatures, we're using DNA polymerase enzyme from a bacteria that lives in hot springs called Thermocycler. And so that DNA polymerase is stable even at a high temperature. The next thing that we'll be adding are free DNA nucleotide bases, so that when DNA polymerase from the TAC DNA polymerase is acting, it has free nucleotides, A's, T's, G's, and C's to add to the DNA. And we will also be adding some buffer and some molecular grade water. So all of that together is mixed into something that we call a master mix. Now I'm ready to add the master mix to my DNA sample. So I have 1 microliter of my DNA sample in this very tiny tube. This is a tube that is going to go in the Thermocycler, this machine to my side right here. And then I'm going to add some master mix to it, 29 microliters. So I will pipette the 29 microliters of master mix. Many of you have seen a micropipetter before, but in case you haven't, you notice me placing a tip onto the micropipetter. We always use one with a tip. And this plunger is set to a certain volume, in this case 29 microliters, to allow me to pipette that very specific amount. So I'm going to push down the plunger with my thumb before I put the tip into the solution. I will lift my thumb up to draw up the solution into the tip. Since this master mix is fragile, I'll put it back on ice. And now I'm going to pipette the 29 microliters into this tiny PCR tube. So you can possibly see this, but it really, 29 microliters looks like about 2 tiny drops. I'm pushing to the second stop on the micropipetter to get that last little bit out. And now I'm going to eject the tip because we change tips each time we use the micropipetter. So I have 30 microliters of solution in here, and I'm going to use the vortexer to mix it. Make sure I get that cap on well. So this is just a way to mix the tubes. You can see it vibrates it a little bit. And so now that it's mixed, because we're dealing with such a small volume, some of the solution could be on the wall of the tube. So I'm going to use the centrifuge to pull everything off the side of the tube down into the bottom. So we learned about centrifuges and balancing centrifuges in our last video, But the key is to just make sure that you have equal size containers that are opposite each other in the centrifuge. So in this case, I have another adapter tube and another PCR tube. And we're using a smaller centrifuge now. We don't need it to go as fast. But basically, we'll go ahead and turn it on, let it run for a few seconds, and then turn it off. Because all we're trying to do is to pull the solution off the side of the tube. Oops. So now that we have our sample here, it is ready to go into the thermocycler. So, whoops, these tubes are a little bit challenging to handle, but that's why we wanted to make sure the lid was on tight. Another trick with these tubes is you want to make sure to label them both on the top and the side, because sometimes when they are heated and there is some condensate, it can interfere with the writing on the top of the tube. So now we're ready to take our sample and add it to the thermocycler so that we can conduct the PCR reaction. So wanted to introduce you to the thermocycler, that it will be heating and cooling the sample. We have the heat block down here. This top part helps to return any condensate down. But the idea is that this sample is going to be alternately heated and cooled to allow the 3 stages of PCR to occur. So you can see that those 3 stages begin with denaturation. So the high temperature will basically pull apart, it'll melt the double-stranded DNA into single strands. The sample will be cooled slightly to allow for the second step, the annealing step, which is when the RNA primers, so our forward and reverse primers, are going to then hybridize with specific regions of our template DNA. So that will allow the Taq DNA polymerase to know where to bind to the template DNA so it can begin copying it. And the last stage is extension. So that's where new DNA strands are synthesized, and that occurs because of the action of the Taq DNA polymerase. So this process of denaturation, annealing, and extension happens multiple times, and each time that it happens, the amount of DNA of this specific region that's specified, that amount of DNA doubles. So by the time we do 30 or 35 cycles, we've got many millions of copies of the region that we're interested in, which in this case is the DNA barcoding region. So I'm going to place this into the heat block in the thermocycler, and we'll go ahead and close that, and then we're going to take a close-up look of what the cycle is going to look like. So we will have the denaturing step, the annealing step, and the extending step. So you can see how our machine is programmed, and then once those steps are complete, the machine will go through each of those heating and cooling steps 35 times. And so after that, we will have many millions of copies of the sequence that we're interested in.